All right, so this is the 2022 paper. I'm just going to scroll down to module 3 quickly. And they will see. All right. This question, this is the 2022 paper. This going to scroll down. So it says, starting from the pollen mother cell, outline the steps in pollen grain formation. They should be able to do it for both the ovum and the grain. So moving quickly, I just pull this up from this study guide. So the pollen grain formation, you just need to draw a cell with one nucleus. I can skip this one and go straight to 4. So one cell divided into 4, then you separate the 4 of them, and you end with this structure. So for pollen grain formation, this is the diagram that you need to know. First, explain the significance of double fertilization. And so that's the info for double fertilization. This diagram here, this says use the information in the flowchart outline how the body controls spermatogenesis. So starting from up here with the hypothalamus, right? GnRH, that is short for gonadotropin releasing hormone. So the hypothalamus will release the gonadotropin releasing hormone. Once that is released, it will trigger the pituitary gland now to release follicle stimulating hormone and luteinizing hormone. FSH goes to this cell of the testes, luteinizing hormone goes to this cell. This cell will produce testosterone, right? Testosterone, once it is produced, some of it will go to this cell, the Sertoli cells, right? Which will lead to spermatogenesis. Now, once you have accumulated a certain amount, the Sertoli cell will, or once this process, it has reached a certain level, the Sertoli cell is going to secrete inhibin. All right. Now, if you notice negative feedback, right? That's a control mechanism. So when this reaches a certain level, you need it to stop. So the Sertoli cell will secrete inhibin. Once inhibin is secreted, it is going to stop the production of FSH. So notice they draw the dotted line to the pituitary gland. So inhibin stops the secretion of these hormones. Notice also testosterone, there's 2 lines, one to the pituitary and one to the hypothalamus, which means that testosterone is going to prevent the production of this hormone, and also it is acting on the pituitary gland as well to stop these. So just to run through again, right? It says outline of the body controls. We're just looking at the control of it. So the hypothalamus secretes gonadotropin releasing hormone. That causes the pituitary gland to secrete two hormones, follicle-stimulating hormone and luteinizing hormone, right? Luteinizing hormone goes to these cells of the testes. Follicle-stimulating hormone goes to these cells, right? Spermatogenesis don't occur over here. Clearly, it is happening in these cells over here. So spermatogenesis will take place. Now, pay attention here, testosterone, it goes here as well. Now, once spermatogenesis gets to a certain level, right, we're looking at the control now. This cell will secrete inhibin. So inhibin is not produced until this gets to a certain level and we want it to stop there. So inhibin will be secreted, which acts on the pituitary gland. Remember, the pituitary secreted these two hormones. So it is going to block the production of these two hormones. If these two hormones are not being produced, then the process of spermatogenesis will end. Along with inhibin, testosterone will trigger the hypothalamus not to produce any gonadotropin hormone. If this hormone is not secreted, pituitary is not stimulated to produce FSH and LH. So to control spermatogenesis, you need to control the production of FSH and LH. And that is controlled by inhibin and testosterone. Because inhibin and testosterone act on the pituitary, both of them, while testosterone only act on the hypothalamus. So that is how it is controlled. Again, this is the 2022 paper. All right, then they gave us a graph. On the y-axis, it has birth weight, and the x-axis is it has number of cigarettes per day. It says, using the information, discuss the effects of maternal smoking on fetal development. So once you look at the trend, you realize that the more cigarette that is smoked each day, the birth rate, sorry, the birth weight is decreasing. So clearly, the smoking is not good for birth weight because the quantity of cigarette that is smoked is inversely proportional. Well, not inverse, yes. The more cigarette that is smoked, the lower the birth rate. So apart from that, some other effects.
You have neurodevelopment issues.
Please repeat.
Neurodevelopment issues.
Neuro, or let's just put that as brain developmental issues.
Spina bifida.
Please repeat that.
Spina bifida.
That's our next condition.
Yes, sir. It's when the vertebral column don't develop properly.
You can spell it.
S-P-I-N-A.
S-P-E.
S-P-I-N-A.
On spinal.
Yes, sir, that one.
Okay. Repeat the condition again. Spinal.
Spin. I'm going to type it in the chat.
No, just tell me so I can just type it here. Quickly. It's spinal and then what? Right, so spinal. Oh, okay. Thanks. PIFIDA. So apart from the low birth weight, these are some other effects. And then now it says, I'm just going to skip the main part of the question. Discuss how two maternal behaviors other than smoking may contribute to low birth weight. So the two other key ones would be, you will talk about alcohol and diet. We'll talk about alcohol and diet as the physician. All right. So you must include one lifestyle change that the physician may recommend for each maternal behavior. So of course, stop consuming alcohol, and they should have a proper diet. Right? So those are the tools that you will actually discuss. And that was module 3. We did that, did this. I showed you the answer for that already. You can look up the function of those. Right. There was an experiment. Let's just look at it quickly. Take the suitable hypothesis. So this one was concerning germination. So a student sets up an experiment to determine the ideal glucose concentration for germination by measuring the percentage germination of pollen grains of patients. Not sure what that word is. All right. Figure 4 shows an illustration of an experiment setup. Each petri dish contains different number of pollen grains with varying concentration of a glucose solution. So state a suitable hypothesis for this experiment. So we're to determine hypothesis quickly to determine the ideal glucose concentration for germination. So we could say as glucose concentration increases, the rate of germination will increase. All right, so state one possible source of error in the experiment. So if we say as glucose concentration increase, the rate of germination is increasing. The key, I was going to mention it, but since I've asked for the source of error, there can't be increasing glucose concentration and the number of pollen grains. If you're increasing the amount of pollen grains, then you're changing two variables. So the source of error, the number of pollen grain was not constant. The number of pollen grain was not constant. So because we're looking at glucose concentration, we cannot be increasing the amount of pollen grain because the more pollen grain, then the more glucose that would be needed. So they should have been using the same amount of pollen grain. State one change in the experiment that could have overcome the stated. So simply use the same number of pollen grain in each experiment. Identify one responding variable. Well, it is germination. So I guess the rate at which a shoot is produced identify one responding variable. So we could say the growth rate or the first sign of shoot. So it's the same thing. Or it could also be the number of pollen grains that actually germinate. So we did that. Coming up to module 2. All right, so in the appropriate, so figure 3 shows 2 of the phases in mitosis. In appropriate spaces provided, make detailed sketches to illustrate the key stages. So we know that which two, so the 2 stages that are missing, it would be, which 2 phases missing?
Metaphase and anaphase.
Right. So you just draw the diagrams too. Let me make metaphase and anaphase. So just Right, so you draw but the cell, right? This time there should be no nuclear envelope, and the cells, not the cell, the chromosomes will be at the equator. All right, so this is metaphase, and also at that point, let's put in our scoop. And then anaphase. So the chromosomes are being separated at anaphase. You know. Anaphase. And so that would be that. Explain how the process of mitosis ensure that each data cell nucleus receives a full set of chromosomes. Well, the first thing that happens is we have the duplication, right? So once you duplicate R, I want to put replication. So you have one chromosome, one becomes two, right? So it duplicates it, and then we know that each chromosome or each cell will get two chromosomes. So duplication, then we basically will have separation of the chromatids from each, it will be sister chromatids from each chromosome. And then each cell will divide. There's like a synopsis of the process. Each cell divide, not each cell, sorry. They're getting diploid cells. So before anything starts, we have duplication of the chromosome. Then once those separate, you will get each cell having the diploid number of chromosomes. Basically,
Sir,
yeah.
I have a question. So if so technically it would have been like in a meiosis one then. Because
Meiosis,
if you have the
yeah,
chromosome
on.
replicating, they would arrange in homologous pairs like meiosis one.
Well, yeah, in a sense, but it's meiosis two that is more, it's a homologous in terms of the homologous
Yes,
thing.
sir. Like if you have replicated DNA, they would arrange in pairs or two homologous pair chromosomes while they are being separated, even though it's one stage. I know that is one
Right.
stage, but I'm just saying, like in meiosis one, you have the first stage of meiosis. They're in homologous pairs. So I'm
Right.
asking if that's the same thing with this.
Yes, so in mitosis, you would have like the two sets, so a big one and like a small one, but we don't need to show the two of them.
Okay, sir.
Yeah, but it would be like that. So this big one here, one would be in this cell, and yes. Third of this one, right? And then a small one and a small one. But it's the same thing that you're basically pointing out. All right, let's just move on there. What else was there? Define the term natural selection. What is that again? If it come back.
Natural selection
yeah,
is the adaptations of an organism based on changes in its environment
right.
to overcome selective pressure so that advantageous genes can be passed on to the next generations.
All right, and you can put it in your own words. So everybody so natural, as I was mentioning, yes, natural and selection, right? So to summarize it up all, right? organisms with favorable traits, and favorable traits are those suited for the particular environment. Organisms with favorable traits, I'm just going to put traits suited for the particular environment will survive, but not only survive, what will they do? Survive and reproduce. However, you want to put this in your own words, all right? So just remember that some organisms will have favorable traits and survive, and the ones without will not survive in a particular environment. Unfavorable traits. And if you don't remember anything at all, just write it speaks to survival of the fittest, or put refers to. So if you can't remember anything, remember it refers to survival of the fittest. If you can remember that it refers to survival of the fittest, I'm sure you can have an idea how to explain what survival of the fittest means. Anybody remember Darwin's observation? All right. So there is overpopulation. So basically, within a species, they should be able to reproduce, to produce large amount of offsprings. All right. So they should be able to populate themselves. There is variation in each species. The population, it remains fairly constant or fairly stable. So there's no drastic change in it. And of course, the offsprings resemble their parent. So offsprings look like parents. Parent. All right, so you will have to use, I'm not going to work this, right? I'm just pause there so I could. So I'm going to clear the screen now if I want to take screenshot or anything. All right. Need to get to the enzyme question and there. All right, so just if I can summarize this quickly. So they gave a scenario, all right? So in pond B, there was a predator, all right? So you put a predator in pond C, there's no predator in pond B. They did not mention what they did to pond A, but just highlight the question so you can read it afterwards properly and assess it. And they want, so you will plot a graph. All right. After that, they want you to use the information, discuss how the results of the experiment relates to variation in the number of spots in the male guinea pigs may support 3 of Darwin's deduction regarding natural selection. So you will use the number of spots. So like this one with the fancy, over time, you realize that it would have the a great reduction in the number of spots, probably due to the predator. So they are trying to evolve to not having any spots, because the spots would make them more visible. Right? But I have to go to question 1 with the enzyme now. All right, so here we have enzyme. This one is pH, this one is temperature, this one is enzyme concentration, substrate concentration. When we did enzyme and their thing, notice there's no tail off, which means that it hasn't reached saturation point yet, so we won't mention anything about saturation. So they want us to use these graphs to explain how each factor affect enzyme concentration. So explain the impact of the following factors using the information. So clearly, as substrate concentration increase, the rate is going to increase. Just have a quick line. Okay, so let's write here. I can put it back here. So at the same point in time, right? The one-time substrate concentration has a slower rate than the 2 times, which is slower than the 3 times. As the increased substrate concentration, the rate is going to increase, right? Until it reaches a point. So if you are referring to the graph, you can just make some mentions. After 10 minutes, the rate became constant. So let us look here. A substrate concentration of products. So if you get the graph, look at the labeling. So it's not rate per se on the graph. So as time increase, the amount of products that is produced increases. So this graph is saying that the rate at which the product is produced increases based on the increase in substrate concentration. Okay. So you are using this graph. So we already know as substrate concentration increase, the rate of reaction increase. We know that, and you can state it. Rate of reaction increase. Then you come to the graph. It's clear that as the concentration of the substrate increase, the rate at which the product is produced increase. So you would mention that. So from this graph, as the time increase, well, not as time increase, you get more products with 3 times substrate concentration than 2 times than the 1 time. All right. So just careful, look at the axis, what is labeled there so you can discuss it. But this statement will always be true. As substrate concentration increase, rate increase. Of course, it increased to a point, then it gets constant. All of them get constant. And at that point, all enzymes are saturated. Remember, when the rate is constant, enzymes are saturated. So substrate concentration increase, rate increase until it gets constant. When it gets constant, it's because the enzymes are saturated. Good. Now, same thing for the enzyme, as enzyme concentration increase, rate of reaction increase. So again, as we increase the concentration, the amount of product that you get increases. And that's because the amount of, you can mention collision once you're talking about rate, all right? So if you increase enzyme concentration, rate increases because collision in both of these, collision between enzyme and substrate is increasing, all right? Temperature. Also, when it comes to enzyme and substrate, the protein, the enzyme structure is not affected. So the enzymes being denatured, it does not occur with enzyme or substrate concentration. That is with pH and temperature. So again, without going into much details, right? Notice this one says rate of reaction, these two. So remember, at the optimum temperature, to the right of it, bonds break, all right? Just remember, bonds that hold the tertiary structure together break, right? So the increased heat, it caused the bonds to vibrate and break. So at this point, just remember that the bonds are going to break. Once the bonds break, the enzyme change shape, all right? Enzyme change shape. Okay. And if it change shape, it will become denatured. On the left hand side, collision, you are increasing kinetic energy. And you should know by now, once kinetic energy increase, movement and collision are going to increase. Kinetic energy goes up, collision increase. If you are increasing collision, rate will go up as well. All right. Kinetic energy increase, collision increase, rate increase. Once you pass the optimum, that increased temperature causes the bonds to break, enzyme change shape. Over here, as the pH is increasing, right? pH increases, rate increase, right? This is the optimum pH. Once you pass the optimum pH, the change in hydrogen ion concentration, so square bracket stands for concentration. So when hydrogen ion concentration changes, that cause bonds to break. Same type of bonds that hold the tertiary structure together, they start to break. You get a different interaction, right? So when the bonds break, the shape of the active site will be adjusted, right? Active site changes, and so the substrate cannot bind, right? So enzyme and temperature, they will change the shape of the active site at a certain point. That is why the enzyme becomes denatured. Enzyme and substrate, they do not affect the active site. All right, complete the table. All right. So plasmids, we know that plasmids are in prokaryotic. Well, not all of them have plasmid. Bacteria have plasmid. So it says, complete the table by indicating whether the following features are present, absent, sometimes present in eukaryotic and prokaryotic. Plasmid, not in prokaryotes, but sometimes. So in bacteria, you have your plasmid. Mitochondria, in your eukaryotes. Remember, prokaryotic cells don't have any membrane-bound organelles. This don't have any nuclear envelope either. And the ribosome is present in both of them. Why am I ticking? You should put present and absent, as it says. Present, 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 absent. Sometimes present. So that would be that. outline the basis of the endosymbiont theory as we have discussed it. So in short, of membrane-bound organelles, membrane-bound organelles, specifically the chloroplast and the mitochondria. Chloroplast and the mitochondria resembling and sharing features of prokaryotic cells. I can jump in some examples. So both, all right, so for example, I can just put it here. Both the mitochondria and chloroplast have their own DNA. What else do they have in common with prokaryotes? They have their own DNA. They have the same size ribosome, 70S ribosome. They are of similar size. All right. So the endosymbiont theory, remember, it shows the evolution or how you go from prokaryotes to eukaryotes. All right. And the evidence for that is the resemblance of chloroplast and mitochondria to prokaryotic cells. I just remember endosymbiont theory explains how prokaryotes developed into membrane-bound organelles inside of eukaryotes. Or basically from prokaryotes into eukaryotes. Remember in this theory, the prokaryote, one large ancestral prokaryotic cell engulfs a smaller one with the ability to either carry out respiration, aerobic respiration, which is the mitochondria now, right? And it engulfed a next one with the ability to carry out photosynthesis, which is now the chloroplast. So it engulfs them, right? And that is how you get the eukaryotes, from prokaryotes to eukaryotes. Right. I have to wrap at this point. Let me see. What was this asking for? All right, explain why the dicot root is considered an organ. So organs are made up of several tissues, right? Carrying out a particular function. So we know that the root, it consists of more than one tissues. This part here, you have the xylem and phloem. I'll bring it up for you. So remember now, with a plant join, you do not show any details. So this is an example of the plant join that you would do. So for example, this, not even so much detail. So you have the circle, and you just do this. I can do it a little bigger. Right. So that's the xylem. Outside of it is the phloem. So with the plant join, you're not given any details of the structures. All right. So notice this doesn't look anything like the actual structure of a xylem. All right. So it's just an outline of it. So on the diagram, you can see here you have the xylem, the phloem, epidermis, and the cortex in the middle, endodermis around it. So for this, You would have the xylem and the phloem. W would be the endodermis around it, and then the cortex. There is none touching the epidermis. So why is the xylem? X would be the phloem around the xylem, which means that look at if they give the root this year, probably in terms of a joint, you can look back at the transverse section of a stem or even the bacteria, right? More than likely, you should get a joint. So you can look back at the stem, the xylem, it's not the xylem, sorry. So the root, the stem, bacteria, sperm cell, just look back at some joints. All right, so xylem, phloem, Z was the cortex and the endodermis. Endodermis. So no any labeling you're doing, know their function. All right. Okay, we did that. I'm going to leave it at that. All right. So I'm going to end it here. Hope this helped in some way. All right. Have a good day. All the best on your exams.
Okay.
Thank you, sir.